Yeah, so my name is Rik Haagmans. I'm currently a postdoc at Amsterdam UMC. The name of our project is Virusinfection and Reactivation and the Gastrointestinal Microbiome in ME/CFS. So the main focus of our project is on investigating a link between gut dysbiosis and the activity of latent and endogenous viruses in ME/CFS patients. So the project only started on the 1st of December. So although I've been working very hard, I don't really have any results yet to share. So I'll just be giving an overview of our project plans for the next 3 years. So the project involves national partners, so we're collaborating with NMCB, but also with ME/CFS lines, but also international partners in the form of Quadrum Institute. So we're collaborating there with Simon Carding, who's he is my former PhD supervisor. And for those who don't know the Quadrum Institute, which maybe many of you, it's an institute in Norwich in the UK, which focuses on gut health, basically. So everything from food science to microbiome research and sort of everything in between. So a little bit of background of the project. So as you probably all know is that many ME/CFS patients experience an infection before the onset of their disease. And there's also plenty of evidence of viral reactivation in ME/CFS. On the other hand, ME patients also very often have gastrointestinal symptoms. There's a high prevalence of IBS, for example. And there's a lot of evidence for gut dysbiosis, so changes in the microbiome composition in ME patients, as well as changes in the immune response to those gut microbes. And crucially, both viral infections and the gut microbiome very strongly affect the immune system. And immune dysfunction is also a characteristic that's very often found in ME patients. So, for example, immune exhaustion, autoantibodies, and things like that. And our project a lot of research has been done on the microbiome and on virus activity within ME patients, but there's not really any knowledge about the link between these two factors, and that's the gap that our project's trying to cover. So we're trying to find out whether there's a link between these two factors in ME/CFS. So the project's based on the hypothesis that gastrointestinal dysbiosis in ME/CFS is associated with an increased intestinal permeability and with reactivation of latent viruses or endogenous retroviruses, which contributes to the severity of the disease. And to investigate this, we have 4 main objectives. So the first one is to characterize the gut microbiome and virome in ME/CFS. Then we'll assess the activity of latent viruses like herpes virus and parvovirus, as well as endogenous retroviruses in ME patients. We'll assess gut barrier dysfunction markers. And lastly, we'll try to also look at causality of the microbiome by looking at the effects of the ME/CFS microbiome components in an in vitro gut model, where we'll be looking at the effects on gut permeability as well as on immune reactions. So the study leverages 3 existing cohorts, basically. So the first cohort is the NMCB cohort. In this cohort, we'll be looking at 70 healthy controls and comparing them to 70 ME patients, half of which will be housebound ME patients. And in this group, we'll be looking at the microbiome, viral activity, as well as gut permeability. And this is sort of our discovery cohort. So this is where we'll compare healthy people to ME patients. And then we'll work with ME/CFS lines to compare their metagenomic data to our metagenomic data to validate the findings in our microbiome analyses. And then we'll move on to the RestoreMe cohort, which is a cohort that's located in Norwich in the UK. So that's managed by the Quadrum Institute. And this cohort consists of patients that are enrolled in a clinical trial. So the Quadrum Institute will be running a clinical trial that's called RestoreMe. And the plan for that trial is to recruit 160 ME patients, which will also have IBS. And these patients will be going fecal microbiota transplantation. and half of these patients will receive a control, the other half will receive an FMT from a healthy donor. And in this cohort, we'll also do the same analysis, and that will kind of allow us to determine whether changes in the microbiome also correlate with both changes in the viral activity in the patients, but also in symptoms, of course, because that's the ultimate goal here. And then lastly, we'll also do some in vitro experiments. So we'll use some fecal extracts from ME patients and healthy controls. And I won't dwell on this for too long, but the plan is to look at the effects of these microbiome components on both gut permeability as well as immune cell activation. So as I said, the project started on the 1st of December, so we still have a long time to go. The project lasts for 3 years. We're currently working with our colleagues at TNO to develop some questionnaires that we need for the microbiome analysis, or to collect some of the metadata. And we're finalizing the fecal sample collection kit, and hopefully we can start collecting fecal samples soon. So I think, with that, I'd like to conclude. So I'd like to thank all our collaborators, both at the research institutes, as well as our patient representatives, both past and present. So the present patient representatives are here on the slide, but also Marcus was a patient representative before, and Kinga as well, Kinga Gumluch. So I'd like to thank them very much for their contributions to the project. So I'm not sure what you mean by epiphenomenon. I'm assuming you're meaning whether it's a secondary effect of a viral persistence. In the case of long COVID, that might actually be the case, but it can also be that that changes in the microbiome also help the persistence of the virus, because it increases inflammation and things like that. But I think we need to do this research to really find that out, I think.